Very good morning to all of you. As a part of the Azadi Amrut Mahotsava 75 years of celebration of independence, ICAR IIRR in association with the Society of Advancement of Rice Research Rajendranagar is organizing webinar in hybrid mode. And today we have our distinguished speaker, Dr. Nese Sreenivasulu with us, and he will deliver the lecture on rice value addition for future roadmap of India. On behalf of the seminar committee, I invite you all to actively participate in this webinar. Now I request our beloved director, Dr. R.M. Sundaram Garu, to give his welcome and opening remarks.
Dear Dr. Nese Sreenivasulu, colleagues from IRR, colleagued from ICAR IIRI, and also our esteemed cooperators from ICRIP centers, and also valued members of Society for Advancement of Rice Research. On behalf of Indian Institute of Rice Research and also on behalf of Society for Advancement of Rice Research, I extend a warm welcome to Dr. Nese Sreenivasulu, who is heading the grain quality unit, state-of-the-art grain quality unit at International Rice Research Institute, Philippines. And he also has another role. He is working. He'll be working as coordinator of ICAR IIRRI collaborative work plan. Particularly, he'll be the research coordinator for this particular program. Dr. Sreenivasulu, for the last several years, he has been consistently working on one topic: grain quality, its various phases, and also improvement of the trait. And he has made significant headway regarding various aspects of grain quality. One of his notable achievements, I would say, is genetically dissecting the trait of very complex trait of low glycemic index. He has identified several genomic regions associated with the trait and has identified or developed improved germplasm. And I think he'll be talking about the progress and prospects of collaborative research in this particular topic. Sir, we invite you wholeheartedly and for this particular talk, and we are indeed very thankful to you for obliging to a request and speaking amongst us. Sir, amongst us we have audience not only from Indian Institute of Rice Research, but also from ICAR National Rice Research Institute, probably from IRRI, and also from several, at least 40-50 All India Coordinated Rice Improvement Project research centers located across the country. And we also have, besides these, a lot of students and also our former staff of IRR, who are also SARR, or Society for Advancement of Rice Research members. I now invite Dr. Arvind Kumar to give a detailed introduction of the speaker.
Thank you, sir. Good morning to one and all. On behalf of Society for Advancement of Rice Research and Indian Institute of Rice Research, I welcome you all to today's talk. It is a pleasure and honor to introduce Dr. Nese Sreenivasulu. So let us begin with his short biography. He got his MPhil from University of Mysore back in 1995 in applied botany and biotechnology. And later on, he went to get a biology PhD from Martin Luther University, Wittenberg, Germany. And after completion of his PhD, he's moved to Germany as a postdoctoral fellow from 2002 to 2005 at IPK. Then he's joined as a project leader from 2007 to 2010 at Leibniz Institute, Germany. And at the same time, he was also parallelly associated with University of Western Australia, Crawley, as a senior lecturer during 9 and 12. And later on, during 10 and 13, he was promoted as a group leader, Leibniz Institute, Germany. During this time, he worked on stress biology, mostly abiotic stress, drought and heat. And later on, he went on to take up the position of head grain quality and nutrition center at IRRI, Philippines from 2013 to 17. And 2018 to 20, he was made the outcome team leader for facilitating rice research through enabling technologies at IRRI. And from 2020 onwards, he's been acting as coordinator for ICAR IIRRI collaborative research. And that is about his work. And some of his areas of research include, as I said earlier, he mostly concentrates on grain quality, like cooking quality, chalkiness, HRR, sensory and nutritional quality. And apart from this, as Director Sir has mentioned, is very much involved with very important trait, glycemic index, and he has done some extraordinary work with related to this very complex trait. And as a group leader at Liebens Institute, he led the research group addressing the molecular mechanisms of stability of seed yield under drought. Now, Dr. Nese has played a very vital role in the establishment of Center for Excellence in Rice Value Addition, SERVA, Varanasi, which was funded by Ministry of Agriculture, Government of India. SERVA basically offers novel tools and support for breeding efforts for all the NAS partners. And it is also likely to work as a network for establishment of value chain pipeline involving all the major players, millers, breeders, agro-industry personnel, traders, etc. And apart from this, it will work and coordinate as the main center for grain quality research for South Asia. And SERVA will also undertake capacity building trainings. And apart from this, Dr. Nese has a long list of publications. I'm not going to mention all that, but some of his publications are in one of the best journals, Current Opinions in Biotechnology, Plant Biotechnology, Trends in Food Science and Technology, Journal of Experimental Botany, Critical Reviews in Food Science and Nutrition, Rice Science, Trends in Plant Science. It's a long list. Actually, he's done a lot of work in biochemistry also. So apart from this, he has been involved in research funding of up to almost $40 to $50 million in both as PI and co-PI. So, and finally, he has been awarded by several international governments. For example, the Vietnamese government has awarded him for his work on grain quality and especially in rice. So with that, I think I offer the floor to Dr. Nese. Sir, please, the stage is all yours.
Thank you so much.
Thank you, Dr. Arvind. Now I request Dr. Sreenivasulu to deliver his lecture.
Before Dr. Nese starts his presentation, I'd like to inform Dr. Swami will serve as moderator for the session, and the rapporteurs or the people who will be recording the proceedings will be Dr. C. Girish and Dr. Shruti. Thank you. Dr. Nese can start the presentation.
Great. Thank you so much, Dr. Sundaram, Dr. Arvind, for the kind introduction and opportunity to be here. It's really a great pleasure to associate with IIRR and basically, create some joint efforts, what we do here at IRRI, as well as the linkages with India. So I have been working in headquarters in Manila, mostly on the grain quality and nutrition program hosted by Rice Breeding Innovation Platform, and also have been contributing at ICAR mostly for SERVA related activities. So today, what I would like to do is take an opportunity to kind of introspect the progress what we have been making together in our journey. And then we should also see what development has happened in the area of grain quality and nutrition, and how we are going to consider this to create value for the value chain stakeholders in the rice sector. So in this particular aspect, what we would like to see is basically India has been exporting rice over decades. From 2013 to 2021, we could clearly see that there is a very linear increase in the growth of rice export, currently reaching up to 14 million metric tons. Mostly that happens in the non-basmati category. So while we fetch around $4 billion from the basmati and the non-basmati, the matching is around $4.8 billion. So most of this rice is actually growing to African countries as well as in Middle East. And we have been exporting like IR64, MTU 1010, as well as Farna under the medium slender types. And also India has been exporting parboiled rice as a source. But much of that export is actually fetching lower values. So what we should be thinking is how can we create premium around non-basmati category, and what we should be doing in the area of grain quality and nutrition to bring those value additional opportunities. So currently the gaps are China and Philippines, they prefer soft texture rice, and India is not exporting much into that category or those target countries. So there is really an opportunity over there to take up that particular program. And also waxy rice is another premium segment which is really being demanded. So if you can bring in some varieties and position in that particular space, that can be the low-hanging fruit. So let's look at the current market segments. So basically the rice traders, they give premium considering 5% broken with low chalkiness, 2% chalkiness, either medium slender or long slender. Basically this fetches around $350 per metric ton. Whereas while meeting that category, if you bring in aroma from India like basmati, it can fetch up to $1,000 per metric ton. Whereas now there is a growing demand, particularly on the lowering glycemic index, because the type 2 diabetes has been increasing substantially in India. And also consumers are demanding traditional heirloom rice because of bioactives present in those pigmented rice. So these are some of the emerging market segments, and we should also think about systematically characterizing the germplasm over there, breeding material, and see how we can position some of the breeding products for those market segments. So broadly, grain quality has been divided into several categories. People are interested about how grain appears based on the length, width, and chalkiness. Then millers are interested about what is really the whole grain recovery. That's nothing but head rice recovery, which is one of the important triad what millers are actually driving, which varieties they're accepting or not. On the other hand, when we cook actually the grains, especially the milled rice, and then what are actually the textural parameters associated with it are also driving the preferences. Aroma, as I mentioned before, is one of the important components which fetches premium. And of course, the composition of starch, protein, as well as bioactives, what is present there can contribute to some of the health beneficial triads, which we need to understand and position that knowledge for product development. So let's kind of look into some of the figures what we have. So we have worked with world rice traders, and we averaged the data and we came up with these figures. When under non-aromatic category between broken rice as well as whole grain, there is $100 difference. While in the case of aromatic rice from Thailand, Thai Homali, as well as basmati from India, you could clearly see that price difference is huge. So almost like twofold price difference. And so if we can take care of minimizing the grain breakage, and then when we add value to the premium triads, then farmers will have more likely chances of getting higher premium. So we have looked around the strategies, in the breeding material, what IRRI has been developing over decades. And we have, when we really look at the head rice yield, this is nothing but the whole grain yield what we get upon milling. So that means the percentage of intact grains which survive milling. The varieties which were released very earlier, they used to have very high head rice yield and an average of 60. So here what we have done is we have categorized all the varieties released in a particular set of decades, and then that's the trend line. And as you could see, we have seen that there is a decline in the trend line of reducing head rice yield, particularly as of now in 2011 to 2012. While chalkiness, which is supposed to be a minimum, but percentage of chalkiness has been increasing. So for fetching higher revenues, what we need is we need the opposite. We need the higher head rice yield and lower chalkiness. So based on this at IRRI, we have now prioritized this as actually a gap triad, and we are trying to work around genetic solutions. So together with our market research team, they have conducted actually some survey in Cambodia, Philippines, Bangladesh, as well as in Eastern India. They have looked into some of the popular rice varieties like PHK, Rumdal, NCRC 222, Swarna, and so on. And these varieties, basically, we asked farmers if we give an opportunity for them to invest their revenues for improvement of certain triads, where they want to invest their resources. Across all other triads, when we look into one of the grain quality triads like head rice yield, very consistently we have seen that particularly in some countries like Cambodia, both wet season and dry season, HRY demand is there, whereas in case of Bangladesh and Philippines, we see as well as India, we see more predominance in the dry season. So that means the moisture content which is there due to sporadic rains and high temperature would play a vital role in lowering the head rice yield, and we should be making all our efforts to improve this particular triad. So when we did not have enough diversity in the breeding, particularly in the long slender and extra long slender, we turned our attention to gene bank, because IRRI hosts around 130,000 germplasm across the globe, and we have picked a large number of lines. We have screened these diversity lines, and what you could clearly see here is actually the green line is the head rice yield. So when we take paddy as 100%, when we dehusk it, more or less 20% is gone, and then when we remove Aliron, additional 10% is gone, and then in any variety we are supposed to get 70% head rice yield, but clearly there is a huge variation. There are some varieties where it's just 10 to 20%, whereas other set of varieties it could reach up to 68%. So when there is such a huge genetic variation for this particular triad, why can't we look into the genetics of this triad and also dissecting the stability of that phenotype? So by doing that, we did identify actually the genetics. So we conducted genome-wide association studies overlaid with transcriptome-wide association studies. We identified a fine map genetic region on chromosome 8 between 25.05 to 25.20 MB, and that has been identified under controlled drying conditions. And we have defined the functional superior haplotypes for the enhanced head rice yield. And then we did identify some of the breeding lines which has a moderate head rice yield, like here the IR64, which has been represented on the left side. If you could see, after we mill it, the broken rice is minimal, whereas head rice yield is very high. Whereas on the other hand, one of the popular high yielding variety, head rice yield is really low, whereas the broken rice what we get is very, very high. So when we tend to look only for the genetic gain for the paddy yield, and if we neglect this kind of an economic triad, then obviously millers won't accept these varieties. So we really need to think about deploying these technologies and fast tracking the development of varieties where this particular triad can be brought in. So here is an example where this functional haplotype, what we have defined for the high head rice yield, we did identify from the gene bank lines, which has been marked in red. And as you could see here, this haplotype, the median in the long slender and extra long slender, we were able to get around 55 to 60. Whereas the inferior haplotypes, what is represented here, like group 3, we noticed that many of the breeding lines were present in that particular pool. So what we should be really doing is going back to our drawing board and then see how we could reintroduce these superior haplotypes in our breeding pipelines and try to enrich those alleles. I think that is one of the strategies what we should be undertaking in near future. And just to give you an example that when we mined the gene bank lines for the haplotypes, whenever we found the gene for the superior haplotype, the upper panel, you could clearly see that, the whole grain percentage is very high. Whereas if that particular gene is absent, then we clearly see that the broken rice is very, very high. So irrespective of the post-harvest conditions, we could clearly see that this genetic region plays a vital role. And we should see how now we could validate these things in the biparental mapping populations, and if possible, position that for future selections. And another important trait which is valued by the industry is chalkiness. So consumers, they prefer the translucent grain, while we see that some of the opaque portions can happen in the grain, which leads to increase in the percentage of chalkiness. And while exporting, the rice traders, they prefer 2% or low chalky grains. And so we were really interested to identify the genetic regions. Again, we have used actually the diversity lines in Indica and Japonica. We have identified a number of genetic regions we named from PGC 1.2 until 12.1. And all these genetic regions, the corresponding PVE values, which are very, very high, sometimes reaching up to 25 PVE, 10% and above, we have identified, and we looked into the epistatic interactions between those major QTL contributing factors. And basically, these are the combinations of QTLs, what we have identified. Many of them have been fine mapped to few genes. And then we went back actually to our breeding material, and then we identified which of those breeding material has which kind of QTLs. As you could see, IRRI 142 has multiple low CHAC QTLs which have been listed over there. And irrespective of the season, we see that being the low CHAC. IRRI 152 likewise has so many low CHAC QTLs. Whereas additional material from 173 until IR40, we have very few QTLs, like 5.3 being the major one, 5.2 in some cases. And interestingly, this single QTL alone itself can actually explain a very low chalkiness. So likewise, we have created a magic population from the high day temperature and high night temperature. And we have screened those magic populations under high temperature conditions. And again, we identified that these QTLs are found to be very relevant for these high temperature scenarios. And again, the chalkiness was very, very low. So obviously, such genetic regions could play a vital role. And we could think about utilizing this knowledge to initiate some of the pre-breeding efforts in future. So besides milling quality, cooking and eating quality plays a vital role demanded by consumers. They are interested about palatability and texture, and that all depends on the type of starch. Although many rice varieties, they do have 90% starch, the amylose amylopectin variation, the cocktail can actually lead to different texture, and also it can impact different digestibility. So what we have done is we looked into the starch structure per se of amylose amylopectin variation, and we tried to identify the right balance where we can convert them into either intermediate or low glycemic index, but palatability should be in the acceptable range. So, and here we have made some specific attempts to do some modeling techniques where we have conducted the viscoelastic properties as well as amylose amylopectin variation across more than 800 germplasm, including the breeding material. And then we have categorized the cooking and eating quality groups into 7 groups. So interestingly, what we could clearly see is, although many of the lines in Indica, they do have intermediate to high amylose like group A, group B, group C, as well as group F and G, they're all falling into intermediate to high amylose, but the model classification, which is linked to the sensory attributes, they are very, very distinct. Like for instance, group A, they would have low tooth pack and crumbly in its sensory attributes. Group B is actually springy in its nature with high residual loose particles. Then basically group C, they will be sleek in its texture. Likewise, group G will undergo retrogradation and they will be dry and hard. So that means the textural parameters which we could not differentiate just based on amylose content, now we can distinguish that based on viscoelastic properties as well as amylose-amylopectin variation. And we can very well link to the sensory attributes, how consumers feel when the taste rise. So these models we now have been applying for the breeding lines. So we can identify which of the mega varieties falls into these groups. And then if a group of breeding lines matches to that mega variety, we can target that and then we can disseminate that material to a particular geographical location. And also that can match a product concept. So likewise, we have made an effort to really systematically characterize IRRI breeding lines released between 1960 until 2015. And as you could see that at IRRI, we have been breeding most of the lines matching to category B2 and category A3. And those are really the lines where we have released in India, as well as part of Africa and Latin America. And particularly in India, like Swarna and Samba Masuri, that kind of a category, which is more preferred here. We have very few lines being identified matching to that kind of a quality segment. And these are IRRI 117, 125, 128, 130, and 171. So we could think about deploying such tools, and if you can position that for the ICAR trials, if you can generate the data and deploy these modeling tools, then we could really identify what material coming in the ICAR trials can be very specifically targeted for certain ecology and in the region. So thereby we would not be able to we would be able to overcome the wastage of resources and rather focus our efforts in utilizing these tool sets. So also to make life a bit more easier, we have been developing some of the artificial intelligence techniques. So these models, the 7 cooking and eating quality models, what we have developed, we have linked that to the resequenced genome sequences. And then we have identified basically 10 SNP markers, 100 SNP markers, as well as 1,000 SNP markers. And then we did identify that this RF models has given us actually the highest accuracy with 0.8 prediction. So we can clearly distinguish using those 1,000 SNP markers, those 7 distinct groups of cooking and eating quality. So in near future, if we can accept that with around 0.8 R-score values, we still can actually go ahead and just use the genotyping data instead of phenotyping. So these are some possibilities where we can think about bringing the high throughputness and try to predict the textural attributes of breeding lines as well as lines what we can undertake for screening. So I want to come back with one specific example. I mean, if you see in southern part of India, as well as in the eastern part, clearly 3 prominent varieties like Samba Masuri, Swarna, and MTU 1010, they are highly popular, and these are the mega varieties. And based on amylose content, we cannot distinguish them. So they are 24, this is 21. Gelatinization temperature is around 75 and 70. But then the models what we have developed and various parameters what we have used for the modeling can very nicely distinguish these three mega varieties, which we could not do based on the routine grain quality measurements. So that's why if we are really keen to develop programs to replace those mega varieties, we could think about deploying these modeling tools in near future so that we could identify the right suitable material targeting each of the mega variety and think about their replacement strategy as we move ahead. So we have come up with an overview where these state-of-the-art grain quality profiling tools, which are available here in India at Serva in ICAR, we could deploy, characterize a lot of elite core collection as well as breeding material. We could include mega varieties and benchmark lines from India as well as from other partners across the globe. And then we could really undertake such a screening efforts for testing the breeding material, then come up with foresighting information, and then link that with policy decision and targeting. So thereby, this kind of a pipeline we could implement in near future, which could be a more targeted approach. And of course, these three mega varieties replacement strategy, we also have been working with some public-private initiatives with Corteva and Syngenta to come up with hybrids which can match to those. And we have some good, interesting insights what we got over there. So likewise, we would be interested about distinguishing the quality into poor quality, medium quality, as well as premium quality based on the overall grain quality measures of milling quality and cooking and eating quality. And we are on our way to bring in some of the tools for selections. So now I would like to turn my attention to the second part, that is health related triads. So quite often, we hear the concerns from the consumers as well as from the biomedical community. When they eat rice, the glycemic index goes up and diabetic patients cannot eat rice. And we have been really looking into what systematic changes have been happening during the domestication, starting from brachypodium until rice. We have measured across multiple varieties and wild species the available carbohydrates. What we noticed is the available carbohydrates we have been increasing from brachypodium to rice, and in rice it's around 74%. And that happened because we have increased the percentage of starch by targeting more of amylopectin over amylose. And then the cell wall, which is a source of dietary fiber, has decreased in these species. Likewise, protein has been decreased, and other nutritional components like bioactives also do happen that way. So now what we need to think is, how can we take care of texture and palatability, but then reintroduce the triads of low glycemic index, plus also the fiber as well as the bioactive compounds which are necessary for maintaining the human health. So we have actually derived the strategy by looking into the biochemistry, that the sucrose which comes into the grain will be cleaved off, and ADP glucose which is produced here, and that is being channeled for making amylose and amylopectin, as well as for the dietary fiber. So the amylose amylopectin, what is synthesized, it accumulates in the plastids, whereas in the fiber related things, basically it accumulates in the cell wall. And then sucrose could also be channeled through glycolysis to produce amino acids, as well as it could produce lipids. So we have been looking into kind of changing the starch structure composition, and also to change the dietary fiber and resistant starch, and to manipulate protein versus starch to an extent. And this puzzle, we tried to address that through genetics. And recently, we have defined actually the genetics, converting very high to intermediate glycemic index, And what we have done is we have screened around more than 300 diversity lines of indica germplasm. As you could see here, the amylose, when it is very low, the glycemic index is very high. But interestingly, when we moved into the region between intermediate to high amylose between 20 to 27, that means the GI varies a lot from high to basically low. And so if we are really interested to capture the low GI, we cannot simply do that by amylose, and we really need an in vitro methodology to screen the glycemic index. So we have made a very substantial efforts to compare different methods of in vitro GI methods, starch composition of amylose, amylopectin fingerprinting, and then we did identify these 3 low GI donors. And then when we went ahead and conducted clinical trials together with Philippine biomedical community at FNRI, we noticed that these 2 donor lines, they found to have a GI of 50 and 51. And we did define the genetics on chromosome 6. We call it as a GI 6.1. Here we did identify 26 loci in addition to GBSS1. And the recombinations, what happens in that region is very, very critical in lowering the GI. So we also validated those donors together with some benchmark varieties. Like for instance, Philippines, they are really interested about soft texture rice. So this Dinarado is a premium quality rice, which is really known for soft grains with higher stickiness, was treated as a control. And then the 3 low GI donors, what we did identify, we conducted the sensory analysis. And we did define actually 2 SNPs. And these 2 SNPs, we could clearly tell whether that's an intermediate GI or high GI. But interestingly, within this intermediate GI, between the GC and GT haplotype, we can differentiate the texture. The GT haplotype would have very high final viscosity. So that means they will undergo retrogradation, whereas the GC haplotype will maintain the texture and will not undergo retrogradation. And interestingly, if you see here, the lines with the GC haplotype, they are basically having a moderate hardness, and uniformity of bite is there, and initial starchy coating also differs. And particularly this particular line matching to that kind of a texture is well preferred over retrograded lines. So I think based on these results, we now have some evidence that we can combine the low GI genetics with the soft texture attribute. So in the beginning, if we can enrich the germplasm for these kinds of haplotypes, we can target the intermediate GI, and then we could ensure the soft textural preferences, whatever is required, and that can be mainstreamed. And on the other hand, we can take up more precise efforts to lowering the GI in the breeding programs. So we did screen the available high-yielding varieties, and then we did identify basically the 2 lines. One is a low GI, the other one is the intermediate GI. So it's in vivo GI, here is 57, here is 55. And basically they are yielding around 5 to 6 tons of yield in the Philippines. In India, the preliminary trial shows that between 5.3 to 5.5 tons with good grain quality preferences. So perhaps maybe we could think about positioning these 2 lines and see whether that could be tested in multi-location trials, and also conduct additional in vivo GI estimations here on the Indian subjects. So currently the low GI market value is around $3.12 billion. And we have identified the gene bank donors, very good gene bank donors, which are from 1 to 3 with the low GI. And then these are actually the high yielding breeding lines. And basically, we could confirm that all of them, they are low GI in its nature. They are medium slender and long slender in its nature. And also they have decent yield so far what we could test in these large plots. So using the low GI, we have been making different products like rice noodles, rice cakes, dosa, and various cookies and so on. Because what happens is when we make flour and then when we process it, the GI goes even much higher than what we use as a whole grain consumption. So particularly for those specialty products, if we can link these low GI and come up with value addition by making the products, that will have a substantial impact. And together with Dr. Leo, our Chinese collaborator, we also have been looking into the strategy to bring in those alleles, what we have identified, both transcriptional and post-transcriptional modification, as well as translational and post-translational modification. And these are actually the important SNPs we identified to manipulate the amylose content as well as amylopectin content. And these currently we are validating to do the genome editing. We are generating genome edited lines in China. So this is maybe another way where we could try to revalidate what we have discovered through genetics and then try to validate the function of those genes. So besides lowering the GI through change in the amylose amylopectin composition, we could also change the resistant starch. And here we were really looking into the genetics for that, and we did find a particular genetic region. This is like SS2A related gene and one more additional gene which was not characterized. Both were found to be very, very relevant. And the haplotype, what we have defined, can really distinguish 0.5% of resistant starch to nearly 2 to 3% of resistant starch. And this is the natural diversity what we can find in rice. But if we can go to some mutants, we could even reach up to 9%. But then texture and palatability will change, and we need to really think about the strategies around that. And together with UK collaborators, we also have been doing the work on dietary fiber, both soluble and insoluble dietary fiber. And here, this is actually the insoluble dietary fiber, what we have characterized for in the diversity and in the magic lines. So while many of the diversity, it has actually extremely low dietary fiber, but we were able to identify some important donor lines and magic lines, which are having a higher amount of insoluble dietary fiber. Those are galactose, rhamnose, arabinose, fructose, and so on, mannose. So these are some potential lines currently we are using to go back and define the genetics for the dietary fiber. And likewise, we have been working on the pigmented rice, and we did identify a novel genetic region on chromosome 7. And this is BHLH and cytokinin oxidase. The combination of that gene, the haplotype what we have defined, has resulted in accumulating higher catechin content. And interestingly, when we test these superior haplotype containing lines versus inferior haplotype containing lines, we notice that the superior haplotype containing lines like haplotype 4 do have higher anti-cancer property. This is for colon, liver, breast cancer. We very clearly see similar kind of trend line. Whereas the rice lines doesn't have this haplotype, you could see the cytotoxicity assay is not at all there. So it is possible that we could really deploy the resequencing data. We could identify important genetic regions, and these donor lines we could utilize to in progress a particular triad in high yielding background. And particularly these anthocyanins, when they accumulate, they vary a lot. Much of that accumulates in the husk and in the bran. and very few accessions do accumulate or retain the bioavailable bioactives in the milled rice. And here is actually one such genetic region what we identified. It retains higher catechin content in the milled rice. And again, this contributed to lowering basically also the GI, because that these particular lines, they have higher amylose 1 and higher amylose 2, and digestibility goes down. And in the previous slide, I showed that the same set of lines do have anti-cancer property, particularly even after we cook the milled rice. So I think these are some important interventions, what we could think about positioning the knowledge, and then see how that can be taken up in the breeding effort. So here in ICAR, we have a Center of Excellence for the Rice Value Addition, where we do have the product development lab and sensory lab. And one of my colleague, Hamida, she has been developing various rice products out of the bran and whole grain from the pigmented rice. And we are trying really to evaluate the nutritional and nutraceutical properties in those rice products. So a way forward would be that right now the problem in diet is we have very high calories and low density of nutrients, and we need to change that mainly in the staple crops. So one way to do that is actually to go back to the diversity, what is there, intraspecific diversity, and tap that, identify the genetic regions, deploy novel phenotyping technologies, including untargeted metabolomics approaches, and then sequencing the genomes, and then linking genome metabolome approaches, and then try really to identify those secondary metabolites, specifically having bioactives, and then validate that them using the genome editing technologies, but then go back to the precision breeding approaches. So on the other hand, if any interesting breeding lines are there, we could work with biomedical community and then start actually the clinical trials, and then establish a pathway for the human nutrition intervention to link all the way up to the gut microbiome and biomarker profiling. So I think if we can do that, we could really change the future dynamics and bring the personalized nutrition to translate the sustained human health. And one opportunity would be we could continue to do what we can by diversifying across the species, but then there is a perishability issues in vegetables and fruits. And so wherever it is possible for us to reintroduce that missing alleles for bioactives and increase in nutrition and lowering the GI, that can be the game changer in future. So I promised you that the roadmap in terms of value addition for India is very, very critical. And I told you in the beginning that currently we are exporting non-basmati mostly on the lower end price, targeting only the broken rice or intact grains in the long grain category, around 250 to 350 US dollars. But then we did conduct some surveys with rice traders, and we did identify that either bringing in aroma or some of the nutritious triads can fetch actually nearly tenfold than what we could be doing from non-basmati with low quality varieties. And so we have to work with not only catering the need of farmers, but work with millers, traders, and consumers, and prioritize these triads like hydrazine, chakiness, which are interested by the millers. Then traders, they are interested on aroma and cooking and eating quality. Consumers, of course, they capture all of that, including the nutrition triads such as low GI, high resistant starch, as well as bioactives. So let's kind of work together and create some opportunities to come up with some specialty rices which could be fitted into that high value proposition, and where at IRRI, we have done some basic work. Clearly, IIRR and ICR colleagues have a lot of knowledge on the ground, and we should come together and look into the current problems, what industry is facing, and then make actually a joint effort to make sure that we would come up with these specialty rices which would have the value addition, and then develop linkages with the rice value industry. So in the introduction, my colleague has mentioned that SERVA's mandate is actually network with partners and then try to make sure that the SERVA will be a relevant platform for support breeding, both of IRRI as well as national partners. And for that, we have brought in state-of-the-art technology here. We have established these labs on food safety, on the GI estimation, on micronutrient heavy metal profiling, mycotoxin, aflatoxins, pesticide profiling, and of course, milling and cooking quality methodology. So we have developed more than 140 scopes, and all these triads right now, we are in the final stage of submitting for APIDA accreditation. And once this is done, then we will be ready to develop linkages with the national partners and see how we can really connect with the activities what IIRR is doing mainly on ECRIP related trials. So if we can be of any help and do some joint activities and be a relevant partners, we will be more than happy to kind of support our journey ahead. So I think in conclusion, I hope I've shown you that adding value to the consumers through grain quality in breeding is very, very critical. And we also created a pathway to enhance the premium to the farmers by adding premium grain quality traits in the background of high yielding lines. And we also need to minimize the barriers for adoption. So still, it's very, very important that we need to think about why we are not able to replace some mega varieties on the ground. What are the best combinations of traits those mega varieties have, and how the new pipeline material coming from our breeding programs can be systematically characterized, and we identify the important material over there, and then we go and target testing in the specific ecologies, and then make sure that the product concepts are positioned. So these are some opportunities, and together we should continue to work to address the UN mandate goals over there. So this kind of a joint work has been done with contributions from a lot of my colleagues, particularly in IRRI, a large number of expertise, geneticists, biochemists, food science team, as well as product development team that have been contributing. And also our leadership, they have been supporting to position these to develop linkages into the breeding programs. So also I would like to acknowledge that we have an active collaboration with IIRR, particularly with Dr. Arvind. And we also have established stronger collaborations with UK, Australia, Singapore, as well as China. And the financial support came from diverse sources from India, particularly Ministry of Agriculture, ICAR. We also started to work with APIDA for the value addition, and of course, other international funding like ACIR, PMGF, BBSRC, CGIR, as well as public-private initiatives. So I would stop my presentation here. I would be happy to kind of address any of your open questions, and look forward to have a joint discussion over there. Thank you, Dr. Sundaram, again for this opportunity to present our results, and really look forward to engagement over the coming years. Thank you.
Thank you, Dr. Sreenivasulu, for your excellent elaborative presentation. Before proceeding to the question and answers, session, I would like to invite Dr. Padmini Swain, Madam Director, NRRI Cuttack, to offer her comments. Dr. Padmini, Madam.
Yes, yes, yes.
Please, Madam.
First, let me congratulate Dr. Sreenivasulu. Excellent topic on value addition as well as specialty rices. And a lot of work he has done and he has presented, starting from our demand, what is our market survey, starting from the gap, starting from the grain quality, from head rice recovery to till the CRISPR-Cas technology. He has done a lot of work, and particularly nowadays everybody is interested for low GI rice varieties, and he has given a lot of emphasis and how to quantify, qualify, and very excellent way. He has done a lot of work, and I think a lot of our colleague also from NRRI and from other participants must be interested to interact with him regarding low GI rice, particularly as it is a demand of the present situation. So better to invite other participants to interact and get knowledge. I have a small query only to Dr. Sreenivasulu, whether they have done any in vivo test for low GI or only in vitro method. As some of my colleagues, they have developed in vitro method and standardized that protocol in our laboratories. And in such way, whether they have done also some in vivo or only in vitro method. And I think nowadays there is a very much demand for in vivo method, and Hyderabad is a good place because National Institute of Nutrition is there, and but our people are not getting scope how to work with human participants. But excellent work he has done, starting from genetics to variety development, that all the quality traits he has done a lot of things, and finally coming to millers, traders, and consumer preferences. With all these parameters, he has done excellent, means giving an excellent talk. I think we should give scope to other participants to interact and get their knowledge enriched, as he has given a very excellent talk. Thank you. I think I do not have much to say.
Yes,
Just
thank you. Thank you, Dr. Padmini Garu. So basically, to answer your question, of course, we did first try through in vitro methodologies to screen the germplasm and identify the donors. And the potential low GI donors, we went back and conducted the feeding trials on the human subjects. We did use actually two different kinds of subjects, the Asian, particularly Filipinos, where we did conduct the in vivo GI trials with FNRI. That's like NIN equivalent organization in Philippines. And the values of 55 and 57 in the high yielding breeding lines, what I've showed, those are actually the in vivo GI values. Whereas the gene bank donor lines, after in vivo validation, we found them to be in the range of one is 50 and another one is around 52. Of course, these are validated, and we had a very good validation between in vitro to in vivo situation. Only one line where it was identified as a low GI from our screening was found to have a median of 57, which is slightly 2 units higher than the low GI, and we are calling that as a intermediate GI. So particularly when we do in vivo GI trials, it's important to have large number of human subjects and independent validations. And the median values what we get, we should take that as average response rather than just making one clinical trial. So, and the second effort, what we did was we did work with Caucasian population in Australia because the subjects also do play some role because there is a difference in the human physiology, particularly on the release of alpha amylase. So we have tested the breeding lines in Australia, and again we found that to be basically the low and intermediate GI in its nature. So coming back, we are having some discussions about how to develop linkages with NIN. And also today morning, I've had some discussions with Dr. Sundaram. So they are already working with NIN, and we will join hands and make sure that these lines we can validate on the Indian subjects. And maybe that baseline data we can then consider as we move forward.
Thank you.
Thank you, sir. Thank you, madam.
Yeah.
Now I request, sir, would you like to?
So some of my colleague also, they are interested. Have some queries.
Yes, madam, we'll go through one by one. Dr. Sundaram, sir, want.
Yeah.
Your presentation was. Let's say your presentation was wonderful. And as the adage goes, one man's food is another man's poison. Same is true with rice grain quality. What is edible for a particular ethnic group may not be edible for another group. And covering the requirements of all the population groups, your group is doing a wonderful job in terms of defining clear-cut parameters related to grain quality. Sir, I have two or three quick questions. The first question is, what is the correlation between head rice recovery and then grain type or grain dimension, sir? Is there any positive correlation? Like generally it is an assumption among the millers that medium slender varieties or short slender varieties, they have got a higher head rice recovery. So based on your study done with a large set of germplasm, is there any such correlation that is found to be true?
Yes, Dr. Sundaram, I think you are right on spot. What we noticed is, I showed you one trend picture. Varieties released in early decades in 1960 and 70. They had very high head rice yield, and then lately, whatever we are releasing, they had moderate head rice yield. So when we kind of look more closely into what are actually the causal reasons, one aspect what we noticed is in the very early program, most of those varieties, they had bold type as well as medium slender type. And then there was a shift in the long slender and extra long slender preference. And most of those varieties, what we are now releasing are matching to that kind of a segment. And so that is one causal factor why there is a decline in head rice yield. The second aspect is, also the temperature and humidity plays a vital role. And the fissures, what it forms, can lead to the development of cracks can be overcome within this particular category. So what we did was, when we noticed this kind of a trend line, when we went back and screened the gene bank lines, we shortlisted only the long slender and extra long slender, and then we started actually to look for the diversity in head rice yield. And I think the genetic region, what I've identified, what I showed, that holds true mostly for the medium slender as well as for the long slender type.
So another quick question. When we talk about breeding for improvement of grain quality, as shown in your presentation, many of these component traits have a high gene to E factor. So keeping this in view, up to what extent, through breeding, we can improve these traits?
Yep. So again, this is a very, very critical point what you're raising, because there is a trade-off between yield and quality. And we need to understand what is that ambient balance which we want to meet. So in that context, recently we have identified one more genetic region which is responsible for increasing the conical branching in the middle to upper portion of the panicle, which could create higher superior grains per panicle. And there we have seen that there was no trade-off. So most of those grains where it leads to increase in the number of grains per panicle because of that upper secondary rachis branches, it had actually a minimum penalty on these grain quality triads. So I think we have some leads over there, and we need to see, I think we need to create more systematic evidence in the mapping populations and test them in different backgrounds. So once we are confident about that kind of a data, then we could probably push for minimizing the trade-off between yield and quality.
Thank you, Dr. Nese. I think we can go ahead with further question.
So any other questions from other participants? Please raise your hand. Sir, please.
Sir, myself is Dr. Udesh Kumar from NRRI Cuttack, sir. Can you hear me, sir?
Yes, Dr. Avdesh.
Please proceed.
Sir, I have two queries. One is that apart from amylose content or resistant starch, is there any other biochemical parameter which is linked with low GI or starch digestibility? And the second one is, sir, should we consider glycemic load estimation along with this glycemic index, sir?
Yeah, again, thanks for those interesting questions. So basically what we have seen is, the first step change, what we need to do is between amylose and amylopectin.
Yes, sir.
So we should target specific types of amylopectin and then combine that with amylose. And we need to minimize actually the extreme short chain amylopectins, which leads to increase in the GI. So that is one proxy trait what we have utilized to enrich the germplasm, where we clearly see a relationship. And on top of that, we can have different strategies depending on what is our end game. So if we want to make sure that we are interested in maintaining the texture and palatability, then in that case, we have to make sure that resistant starch should not increase too much. So we have to target ambient levels, including the dietary fiber, what is present in the endosperm. So, and in case of pigmented rice, we would have the possibility to get the flavonoids or anthocyanins, which are accumulated, can also contribute to lowering the GI. So there are different puzzles, and depending on the product types, we can design the breeding concepts to lower the GI.
Sir,
coming back to your, back to your second question on whether should we consider only the GI or we should consider the glycemic load. So ideally, nutritionists, what they recommend is they recommend we need to consume only 1 cup of rice per meal. So, and as long as we ensure that consumers, if they stick to that recommendation, and then if we are bringing in the low GI triad, we take care of glycemic load. So the problem comes when we make very delicious diets, and then we cannot control ourselves and we end up eating more, right? So that is where glycemic load will increase. So we just need to create more consciousness among consumers. And I think that's the only way how we can make the difference in terms of glycemic load. Thank you.
Thank you, sir.
Sir, I have one question. Do you have any breeding program for a targeted amylose content, say low or high? Sir, am I audible, sir? Sir?
Can you repeat again? Can you repeat again, please?
Sir, do you have any breeding program for a targeted amylose content, say low or high, like that? Amylose content. For amylose content, do you have any breeding programs?
Sure. I mean, most of our product concepts are derived based on amylose content only. So at IRRI, under one rice breeding strategy, they have defined 9 breeding product concepts. Out of them, 7 are being mapped for India. So I think in that they are targeting actually intermediate to high amylose types. Whereas for Bangladesh, they are looking more of high amylose types. And if we are looking into the market segments in Southeast Asia, they prefer low to very low type of amylose content. So as I showed in my presentation, even if we target intermediate to high amylose, if we are interested to capture the right textural attributes, amylose content per se is not sufficient. So this is where I think we have to think about the modeling techniques to find out varieties which matches the right textural attributes, and then that can be a good mechanism in future for us to utilize those tools for future equip related trials. I mean, this is just a preliminary thought. We should have more engagement of discussions on the topic.
Dr. Nese, I have a quick question. Has any group, including your group, done any research work on grain quality attributes of different wild species of rice? Like what are all the typical attributes of representative accessions from belong to each wild species of rice?
Yeah, unfortunately, we haven't done much on the wild rice species characterizing the grain quality. We do have a scientist, Dr. Sungrul. He took over from Dr. Jana and he's continuing the development of material. So basically, that is part of our plan to analyze for the quality parameters as well as for the nutritional parameters. But so far, material has to be progressed that is in the pipeline, and we do not have any experiments as of now.
The reason why I asked this question is there could be novel sources for some of the niche traits which may have a significant market value. That's why I asked this question.
Yeah. I think in Rufi Pogon, there is really a possibility. I mean, people have identified that that can be positioned for a particular type of food priority. And also there is a variability in terms of cooking and eating quality. So Dr. Rodwink is interested on that, and we have discussed. But yeah, we need to wait until they fix all the materials, and then we can take up the characterization.
Yeah, Nese, this is Dr. Balram speaking. Actually, you said that low GA is the highest priority as of now, but the breeding is not going to the level what is required just because of the technology, laboratory technologies wherein in vitro evaluations are not matching with the in vivo, right? So, are there any, progress in identification of any surrogate traits or something where improving the in vitro methodology as of now to match with this in vivo techniques, because the in vivo is the very complex and time-taking process that is delaying all the research work.
Yes, indeed. I think that is a rate limiting step for any breeding program, right? So if we do not have the right phenotyping methodology, which is not high throughput, we cannot take up any breeding program. And this is why we could not really progress much in terms of breeding the low GI. Rather, we have screened some germplasm, and maybe we identified few lines, and then we tried to position those lines as an early outcome. So what we did was we have made actually a very substantial comparison between various in vitro GI methodology. So the way how people they are using as a starting material, whether it's a flour or granules or whole cooked grain or milled cooked grain, that itself can lead to a big variability. Then the second is the cocktail of enzymes and the amount of enzymes what we use for in vitro digestibility plays another significant role. So in the literature, there was a lot of confusion and those methodologies could not be compared to one another. So we have actually made a very substantial effort in the very beginning to compare different methodologies, and we have standardized. And the methodology what I showed you, where out of 6 lines what we have identified as a low GI, 5 lines we were able to validate using in vivo GI. 1 line median moved to 57 instead of 55. So you could imagine that this methodology has a more robustness to at least compare with in vivo situation with Filipino subjects. So we haven't done much on the Indian subjects. So definitely we will take up those initiatives as we move forward.
Dr. Seshu, you have raised your hand. Dr. Seshu Madhav.
Yeah. Thank you. Dr. Nese, very good presentation. I could see a lot of leads on different aspects on HRR and GA. I have one query on GA. You said your genome-wide association and also transcriptome-wide association. You said the chromosome 6 has a 26 loci, but you said that GBSS1 haplotypes are giving some kind of a clue for your low GA lines, whatever you shown, three low GA lines. Are other loci also have a inferior haplotypes in those genotypes? Could you throw on that?
Yeah, thank you. So what I really like to clarify is that GI 6.1 region, we identified that important SNPs, what we identified in that particular genetic loci has 26 genes. And those were found to contribute actually to the highest PVE values for lowering the GI. So when we consider only GBSS and the variation around GBSS, there we could easily manage between distinguishing the varieties between very high to high as well as intermediate GI. So, but for lowering the GI below 55, alone GBSS will not be sufficient. We need other recombinations happening downstream of GBSS. So that is currently work in progress, and we are looking into not only defining the genetics for the low GI, but also for validation of those genetic regions.
Yeah, I have one more quick query. Yeah, sorry. Yeah. Thank you. Dr. Nese, that means that there are indeed some genotypes where your GA is low, where other genes in that loci have a different haplotypes which is favoring those low GA.
So yes, of course, the GBSS together with other genes, that combination is critical to bring down from intermediate to low GI. So that is the work in progress as of now.
Okay. Another quick query, sorry. We have varieties from our own institute like Improved Samoma-Suri, Lalat, Sampada. Those ones which you have any data on those, what are the haplotype?
Yeah, so in fact, like, we could not actually get that germplasm to IRRI HQ because of restrictions. So now we have brought those technologies in Varanasi, and we have been standardizing those methods here. And definitely this is the right time to identify all the important checks, and then maybe think about generating the data with the methodology what we have. So that will be an useful exercise what we should take up.
Thank you.
Dr. Subhasisa Bal. Dr. Subhasisa Bal.
Good afternoon, sir.
Please.
Yes.
Please.
Sir, I have a question to Sreenivasulu, sir. Sir, we have a lot of variety which is having a low GI. like Swarna, ISM, BPT, Biridhan 29, Lalat, Sampada. These are having, these are coming under low GI rice. And exactly right now I'm working in the ground level, exactly in the field. I came back from the IRRI and I came back, I am staying in my village. There I am working with rice. So I'm facing problem. So as an entrepreneur, I'm getting problem to sell my rice. Exactly whenever I'm talking about these rice having the low GI in front of a customer, accepting, they know they have the diabetes, they have all other things, but they need the 25 rupees of rice. If we'll go for these rice, they will not accept these rice. There is a gap, and farmers are not interested to cultivate these rice. Some of farmers are cultivating, but majority they will go for the high yielding variety. One character they will choose only high yielding. This is the first question. Second question, I have some lines. Sir already talked. The lines having purple in color, exactly a presence of anthocyanin. I have some lines. Those are having anthocyanin content in the root, in the leaf, in the grains, in the husk. So that line I want to share to either DRR or NRRI or to IRRI, because it will be helpful to the scientists to develop some lines regarding low GI rice. So if any scientist interested, I can share that particular lines. I have collected during my PhD period. So I have collected these rice from the different part of the Odisha. So I want to share this rice so that it will be beneficial for the society.
Yeah, thank you.
Dr. Subhashisha, yeah, thank you very much. And we, along with NRRI, will certainly be interested to take forward these lines, and then eventually we'll share it with Varanasi Center of IRRI also. But regarding your first question, my expectations are very contrary, because different from the market in your village, here in Hyderabad, if a variety is simply labeled as low GI, we are seeing, like, say, there is a very good local variety called as Telangana Sona. It is selling like hotcakes, even at 100 rupees, 90 rupees also, they are marketing, and then it has been clinically validated. And I think the farmer, when, like, say, it's a work in progress, I think you need to make some more efforts to make convince them to go for cultivation of high value varieties like low GI variety. It's my humble submission. Am I right, Dr. Nese?
Yes, indeed, Dr. Sundaram. I think what we really need to do is, like, as long as there is no penalty between yield versus these value-added triads, then farmers would be happy to kind of test and accept that. And then we need to develop market linkages. So create awareness around and bring value chain stakeholders, and then create a premium around. And once that happens, then naturally farmers would be willing to go for those specialty triads. So I think the variety what Dr. Sundaram has mentioned is actually the best example how one could try to create awareness and bring stakeholders to make that happen.
Chat box. Sir, we have some questions in chat box also. I'll read a few of them, sir.
Sure.
Yeah. So, Mahesh, sir, here the Dr. DVK and Rao want to inquire whether there is a documentation of different grain quality of rice grown in different soils. Element, that is, T5 elements come from soils, while a plant needs 18 elements to complete its life cycle, emphasizing the role of soils.
Yeah. So, I mean, of course, the soil health and nutritional content of soil is so critical. But in terms of grain quality, at least like routine triads, we did not see that that is being impacted majorly. So what if we are interested more on biofortification strategies there, then it becomes very important that we have to map the nutrient content in the soil and then see whether some of these biofortified varieties what we are growing can be, whether that can be grown in the soil which is rich in nutrients.
Arvind.
Yeah, Dr. Muthraman has a question.
Sir, congratulations, Dr. Sreenivasulu. I have been hearing your lecture last 10 years, or ever since you joined in IRRI, whether it is in Raipur or Hyderabad. You are a permanent fixture in our agri workshop. Very interesting lecture you have delivered, sir. My first question is, your entire lecture was, your gamut of lecture was divided into 3 parts. First part, I'm raising one question, sir. Other than the imagery analysis, is there any standard objective method to classify the rice varieties, sir? Am I audible?
Yeah. So you mean to say that for the size and shape?
Yes.
So for the chalkiness and for size and shape, mostly we rely on imaging technologies like videometer data is actually one of the technique what we are currently using, and also Vibe is another instrument which is again imaging based technique. So those 2 techniques we can really handle large number of samples simultaneously. We can score grain size and shape as well as the chalkiness, percentage of chalkiness.
Okay, sir. My next question is, you have mentioned about the chalkiness is directly correlated with the translucent in rice. In case of colored rice or Kauni rice or black rice, what is the percentage of chalkiness? It is not translucent. That is maybe one reason why the consumer acceptability is not there. How to improve upon it?
Yeah. So, I mean, in case of pigmented rice, said we cannot easily estimate the percentage of chalkiness because of that, spectrum limitation. But currently, we have been looking more on Vibe related technique to distinguish different levels of bioactive compounds present in the bran, whether that can be linked to the antioxidant potential. So in case of pigmented rice, chalkiness will not be critical. Only in case of milled rice, especially the translucency would be an important measure. Whereas in pigmented rice, consumers won't treat that as a requirement.
My third question is, you have mentioned in your table, you have studied, analyzed the varieties from Cambodia, Philippines, Bangladesh and Eastern India. Sir, actually, any analysis your lab carried about the Boro rice, because it is a long duration rice. From flowering time to harvest, the temperature will go very high. In spite of that thing, it gives a bumper yield. So how that high temperature is affecting the chalkiness, sir, in Boro rice? Any analysis carried out, sir?
Yeah, actually, we have one project implemented, in Bangladesh as well as in Myanmar, particularly targeting the high night temperature regions. So there, some of these donor lines in the breeding with low chalkiness, what I showed, validated based on the QTLs defined over there. We have tested in those hotspots and then we found them to be consistently low chalk. So that is why this 5.3 region, what I listed over there, could be a very relevant and important genetic region for low chalkiness.
Yeah. Now, next one is not question. It is some vested interest. I have a big team with me. If you want to do any consumer analysis and all that thing, we will be willing to collaborate with you. Already Dr. Nirmala from my department, agricultural economist, is doing, sir. If southern part any such work has to be carried out, we are willing to extend our support, sir. Thank you.
Sir, Dr. Padma Kumari has Dr. Padma Kumari, please.
Sir, good afternoon, sir. Thank you for a nice presentation. I have one query. I'm an entomologist, Dr. Padma Kumari, sir. Here, usually 5 days, 5 decades ago, farmers used to take rice soaked in water previous day or 2, 3 days, add lemon leaves, and they used to take it and they go to the field. They were contented with that, and they used to stay for the whole day. And nowadays, rice is not recommended as a breakfast cereal at all. So, what are the biochemical changes that have been occurring from the days we developed yesteryears and now? And now the present day rices, if you cook, the keeping quality of the cooked rices is very low. It just it becomes smelly and it will not stay for the whole day also. So, how would you see in future about the keeping quality of the cooked rice and rice as a breakfast cereal? Do you have any comments on this?
Yeah, thank you. I think that's very, very critical. I mean, if we see farmers, that the most important aspect, what they are really keen is about whether they can use the overnight cooked rice for early morning. That percentage is very, very high. And just to give you an example, why Swarna and Samba Masuri, they are so popular. Besides having high head rice yield, this is actually another important trait where farmers, they like it because the spoilage is not there. And mostly these are very firm grains, and then retrogradation would happen. But then once we add water and cook it, we are able to bring back that desired texture and it won't spoil. So I think that's a very, very important aspect from a practical perspective. We haven't done much on that space. Definitely, we need to work together on such an important problem. So coming back to the products for the breakfast. So at Sarva, we have been looking into the possibility how we can deploy either kala namak or pigmented rice and some of the specialty rices for making products like we recently found Samba Masuri is found to be very suitable for making popped rice. The volume expansion is very, very high. While we look into some of the pigmented rice, that acceptability range is there, but then other nutritional content retained at a higher level. So I think these are some opportunities where as part of a value addition, for developing rice products, my colleague has been working in that space and she has been prototyping various rice products. So once we make progress, I will be happy to kind of report back. Thank you.
Sir, we have one question in chat box. How GA differ between waxy soft rice and non-sticky rice?
Yeah. So I mean, I showed you in one of my graph that particularly when we have waxy and very low amylose type, then generally all those lines will be very high in its GI. And then when we move towards intermediate to high amylose, there is a variability ranging from low to high. So clearly one of the reasons why in Southeast Asia, while they prefer the soft textured rice, they have this problem of having very high GI. Whereas in India, we have actually high to intermediate in its GI. Of course, it differs in terms of textural attributes as I shown with a 2 SNP combination.
Sir, another query from Dr. G. Padmavati. In view of changing climate, particularly temperature, drought, salinity, and submergence stresses are more prevalent, affecting adversely the rice quality, grain quality parameters. I would like to know the status of grain quality breeding efforts when subjected to abiotic stresses in rice. That is our question, sir.
So, yeah, I think under Strasa, we did some work about looking into the impact of different stresses on grain quality. We noticed that drought and salinity, basically they impact negatively the head rice yield as well as chalkiness. whereas amylose and all, they were more stable, just a few units do change. And interestingly, under high temperature, we noticed that even the amylopectin varies quite a bit. So these are some implications. Again, not all stress will impact similarly. We need to invest more efforts to understand comprehensively what happens in each type of a stress.
So there is another query from Dr. Jaiprakash Aditya. Sir, with the same amylose content, suppose intermediate cooked rice texture, in case of which rice, brown rice, red rice, or pigmented rice, rice are turned to be different, and cooking turns also differ. And what are the major cause and how it can be reduced?
Yeah, of course, that what you are indicating, red rice, black rice and all, if it is consumed as a whole grain, basically that will have the dietary fiber source, bioactives and all that can vary the glycemic index, although they would be intermediate amylose, but additional dietary fiber and bioactives can actually lower the GI by a few units. While when we consider basically the milled rice, then we take away the dietary fiber and bioactives, and then for mainly texture and palatability purpose. And there we need to make sure that subtle changes what is required to achieve the low GI is mainly through change in the amylopectin composition, and of course with insoluble dietary fiber plus some resistant starch can play a role.
So Dr. B.M. Singh from Gorakhpur wanted to know whether you have analyzed Kala Namak for GI and other quality traits.
Yeah, no, we haven't analyzed yet for the GI in Kala Namak, but we characterize Kala Namak for grain quality, micronutrients, including the cooking and eating quality. So that is currently under plan. So soon we will have the data on the GI.
Sir, aroma or grain type will influence the GI amylase content in rice. This is an inquiry from Sunil.
Yeah, no, we haven't seen any such relationship between GI and grain type or GI and aroma. So that's not relevant.
Sir, with this, we finished the queries in the chat box. If there are any more queries from the participants.
We have one more duty to do.
Yes.
Vote of thanks.
Sir.
Sir, you can join us in the committee room, sir. Dr. Nese, you can log down. We have one more thing to do. Yeah. No, we can call him. Sanjeev is there. Yeah. So I'm requesting all the participants to send a list of their questions in the chat box or through email. We will send it to Dr. Nese, and then he'll provide you the answers. Sir, on behalf of Indian Institute of Rice Research and then also Society for Advancement of Rice Research, it's a proud privilege to felicitate you, sir. So we should have started with a bouquet initially for welcoming you. Some more reasons, sir. Thank you. And we would also like to Shall we go ahead, man? Yeah. You can just ask any questions.
If there are any more
questions.
Hello. If there are any more questions, Dr. Sreenivas will answer. If there are more questions, I will proceed further. If there are any more questions? Chat box, put Mahesh, chat box.
There is no new questions.
So there is no more questions. Then I request Dr. A.P. Padmakumari Madam to deliver vote of thanks.
She's chairman of our seminar committee. You take this one. Okay. I think batteries.
It is there. Thank you, sir. Respected Director, our distinguished speaker, Nese Sreenivasulu, Research Unit Leader, Consumer Driven Grain Quality and Nutrition, Scientific Fraternity of IIRR, NRRI, PJTSU, NGOs, and other state universities, students, ladies and gentlemen, it's my proud privilege to propose the formal vote of thanks for this webinar. First, I thank our director for the initiation of the seminars under the aegis of the Azadi Amrut Mahotsava and our co-host for organizing all these seminars, SAR, and its team, headed by the General Secretary, Dr. R.M. Kumar. I profusely thank Dr. Nese Sreenivasulu for accepting and delivering this talk in this 12th webinar in this series. The topic gains importance in the current trends where quality versus consumerism and marketing play a major role in generating revenue. In this talk, sir, you have walked through the whole gamut of quality parameters, importance of this trait for the farmers, the millers, and the consumers, and also emphasis on the various approaches for increasing the value of the final product. And you have stressed on the integration of the amylose and the importance of the texture, and finally the significance of modeling in delineating various other parameters to arrive at the final conclusions with regard to various quality traits. I also thank Dr. Padmini Swain, Madam Director, NRRI, and Dr. B.N. Singh, former Director, for joining this webinar and participating actively. I thank Dr. Jukanti, Dr. Evia Swami for conducting this webinar, and Dr. Shruti and Dr. Girish for recording the proceedings. I thank each one of you for sparing your time to participate in this webinar and making it a very great success. And I thank Dr. Sailaja and her team from AKMU for generating all the logistics for the conduct of this seminar, and the social media committee for the popular communication so that all of you could participate actively. Thank you, one and all.
Really nice.
We'll end this meeting now. Thank you. We declare this meeting closed.
Now this meeting is closed.